All right, morning, everyone. So, a couple of quick things. Remember, there is a Brightspace quiz that is going to open up after class today. So the lecture quiz is worth 15 points, 35-minute time limit like it has been for the semester, stays open until 5 p.m. tomorrow. I will email out a reminder about that then as well. There is a interactive notes lecture set that is due then on Friday. So Friday there is then which one did I say? The microbial genetics interactive notes is then going to be due on Friday. That was the one that we just kind of finished up the last little bit at the beginning of Monday's class then. The quiz will cover a mixture of questions, so that's the interactive notes. One other thing, remember the quiz is going to cover stuff from the last week of class on things. Could be some DNA replication, PCR, gel electrophoresis, genetic transfer questions, and some other stuff that we might talk about in today's lecture. Questions about either of those two assignments that are due this week? Today we're going to be lecturing. Friday we're going to be lecturing. Monday we're going to be lecturing. Remember then, exam number 3 is coming up on then this coming Wednesday, a week from today. So exam 3 is a week from today, will be the same format as previous ones. I will be getting the pathogens of interest study guide posted sometime later today on Brightspace and emailing out about that then as well. But it is going to be a mixture of multiple choice questions, possibly some matching questions, the pathogens of interest, and then it will have a short essay question or two then on as well. So same format as what we've been going through. Some students do better on the multiple choice. Some students do better on the essay. So I usually like to have at least a little bit of a mix of both. Okay. Questions for the exam 3 then? Also remember that Friday, April 18th, next week's Friday is Good Friday, and we don't have class on that day. So we do have class this coming Friday in a couple days, but next week you have a long weekend break. If you did not pick up your exams last time, then let me know. I did end up having a student bring an issue with exam number 2 to my attention. There is I messed up the answer key on one question for anyone with exam 2 version 2. So if you had the first version, and I'm going to send out an email about this, there's not a good way for me to handle this situation since I handled back the exams already. But I made a mistake and filled out the wrong bubble on the answer key, basically. And so on question number 28, the answer key said it was D when it was supposed to be A. And so if you have the first version of the exam, no problem whatsoever, and you don't need to do anything. If you have the version 1, it doesn't say it's any version. I'm going to send out an email with better instructions on this and some pictures. But if it doesn't say any version number on the front page of the exam, you had version 1 and you don't need to do anything. If your exam reads instead, it goes your name, and underneath that it says exam number 2, 100 points total in parentheses, and then it says V2. Well, what I would like you guys to do is bring your exams to me, and both the exam copy and the Scantron copy, and I'm going to take a look at your question number 28 to see if you need points back on it. Okay. There's no good way for me to handle that other than that. So you guys do have to have your copy of the exam. If you've picked it up already, you have to come bring that to me after class, and I'll get names copied down. I do need to see the physical copy of the exam if you're to get points back on it. You only get points back on it if you did have the correct answer choice then. Does that make sense, everybody? So it has to be the A. So that's how I'm going to handle that one because that's what it should have been. Questions? I'm going to send out an email with the instructions on this then as well. And also so that people that aren't here today have access to that information. Okay. So we'll do that after class though, please. So again, if you have version 1, doesn't have any version number on it, don't need to do anything. If you got version 2 and you answered A for question 28, bring me then the exam after class. If you guys don't have the exam or you don't come bring it to me, then that's just going to assume everything was fine. Okay. All right. All right. So today though, we're going to keep on talking about then horizontal gene transfer. We left off talking about transformation and conjugation last lecture. So transformation, this is any organisms, any bacteria that have the COM machinery are capable of transformation. The COM machinery is an enzyme complex that they're going to use to take up any free-floating DNA out in there from their environment. So there's DNA floating out in the environment. Bacteria with the COM machinery will take it up, and this by the COM machinery, and that process is called transformation. Those bacteria, if they take up DNA, sometimes they gain any genes that were coded on those DNA segments then. And that might be a gene that controls some type of trait they did not have previously, like for instance, antibiotic resistance then. Maybe there was a gene that made the bacterial cell resistant to antibiotics. Bacteria recipient came along, it was not antibiotic resistant, but then it picks up that gene and it gains the ability to be antibiotic resistance because it picked up that gene, for instance. We're going to talk about some examples of this as we go through the remainder of the semester. Conjugation is a different type of horizontal gene transfer. This is where then any bacteria that have what is called an F factor positive plasmid. Plasmids are small circular pieces of DNA. There's a lot of different kinds of plasmids. An F factor plasmid specifically has an origin of replication, codes for genes that will make a pilus, a sex pilus, and then codes for genes that will make the TRA machinery, a DNA transfer enzyme complex. So it has to have those three things on the plasmid to be an F factor version of a plasmid. But this will export DNA out of a donor cell and into a recipient cell then. And we talked about that one last lecture. Today, we're going to start off talking then about the third type of horizontal gene transfer, which is then transduction. And this is transfer of genetic material from one bacterial cell to another through a bacteriophage, which is a type of virus intermediary then. So the virus, the bacteriophage, will move DNA segments from one cell to another different recipient cell then. And there's two different types. There's generalized transduction versus specialized transduction. Again, in transduction, what is happening is a phage or bacteriophage. This is a kind of virus, and I'm going to talk about more in just a second, but a phage is a type of virus. They accidentally package up bacterial chromosomal DNA sequences. They're not supposed to do this. This is a mistake by the virus. And then they transfer those bacterial DNA sequences to a new, different bacterial cell. So it picks up DNA from one cell, the donor cell, and then the virus moves that bacterial DNA from the donor cell into a new, different recipient cell. There's two different types of transduction. There's generalized versus specialized transduction. In generalized transduction, the mispackaged bacterial DNA can come from any general location on the bacterial chromosome. It's not specific, it's general then instead. Specialized transduction differs in that the DNA, the bacterial DNA that is packaged up and transferred, can only come from specific spots, special spots in the bacterial chromosome. And it has to come from a part of the bacterial chromosome that is adjacent to something called a prophage integration site, which we're going to talk about here in a second. I'm going to go through generalized transduction first, and then we're going to go through kind of the steps that are involved in a specialized transduction event. Okay. So this is kind of the difference. But again, something called a prophage is always going to be involved in specialized transduction, but not in generalized transduction. And we're going to talk about what that prophage is here in a second. All right, first off, bacteriophages. We've been using phage or bacteriophage. Those two terms are somewhat interchangeable. Phage is just a shortened version of bacteriophage. A bacteriophage is a type of virus that specifically infects bacterial cells as its host organism. So for it, bacteriophage infect bacterial cells and only bacterial cells. So these are types of viruses that use bacterial cells as hosts, but for instance, would be harmless to us. Most of what we know about viruses is from studying bacteriophages. They're safe to work with. They're similar to other kinds of viruses, but they don't cause disease in humans. Once viruses in general, though, they infect a host cell. When they infect the host cell, then they use that host cell to make copies of themselves. Copies, 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 copies. Phages and viruses, they lack ribosomes. They cannot process their own energy. So they can't do energy processing. Because of that, if we have a virus just by itself, it can never reproduce. Viruses, there's a lot of debate on if viruses are true living organisms or if they're not living organisms. Most biologists classify viruses not as living things, but as like complex molecules, then in essence, complex molecular machines instead. Because again, by themselves, they cannot process energy. By themselves, they cannot reproduce. They have to infect a host cell to reproduce, and they hijack parts of the host cell. This also makes them obligate parasites. What's it mean to be obligated to do something? You have to, and viruses have to be parasitic of a host cell or disease-causing then of a host cell. They have to infect a host cell, they hijack and take over the host cell, and then they use that host cell to make copies of themselves. In essence, it looks a little bit like this. This might be in blue. In red, this is going to be my viral particle. So this guy right here, this part right here, this is going to be my virus or my phage. This other part right here, again, is going to be my bacterial cell. So here is my bacterial cell, and this is going to be the bacterial DNA or chromosome, the circular chromosome. This is going to be so again, this is going to be the bacterial DNA chromosome. In red is then this guy in red right here. This is the viral genome then instead, the viral DNA. What's going to end up happening is this phage kind of comes along. It docks with the host cell. And so it docks with the host cell. So here's my virus. It comes along, docks with the host cell. And then basically what it does is it injects its chromosome, its genome, its genetic sequences into the host cell. Sometimes it also injects a few enzymes along the way. This is kind of just my way of showing that. But also right over here, we kind of just see this viral genomes in red. The virus particle docks with the host cell and then it injects its genome. This is actually an electron microscope image showing that process. These are some phages. And here is the viral genome, viral DNA getting injected into the host cell down here. So we can actually see this process physically happening via microscopy in some cases. It sometimes also ejects not only the phage chromosome, but a few enzymes along the ride then as well. Once it gets inside of there, most viruses, what they do is they actually digest the host chromosome. So they take that host chromosome and they just cut it into fragments. At this point, the host cell is dead. It just doesn't know it yet. What you kind of have left over is a zombie host cell. So the DNA is all cut into fragments. When a DNA of a cell gets cut into fragments, that's going to be lethal to the cell. But inside that cell is still ribosomes. There's still ATP, NADH, proton motive force, for instance. There's still nucleotides, amino acids, DNA polymerase, RNA polymerase, ribosomes. There's all that stuff is left over there. And the virus takes it all over. The virus starts using host cell ribosomes, host cell DNA polymerase, host cell RNA polymerase, to instead of making host cell stuff, to making viral stuff. So then the virus hijacks all the host cell materials and nutrients and starts making copies of viruses inside of it. Makes many, many, many copies of phage chromosome, many, many, many copies of phage proteins. And eventually, it starts making mature, intact, new viral particles. All that stuff assembles together into a new virus. And eventually, then those viruses, those phage, burst out of the cell, and then they go off and repeat this cycle, infecting new host cells. Questions about that? Now, typically, when this happens, you are inside that host cell. Remember, you're making copies of the viral DNA in red. You're making copies of my phage proteins here that are being shown. Usually what happens is those phage proteins, those viral proteins, will wrap around a copy of the viral genome, the viral DNA, and then you have an infectious phage or infectious virus. So almost all of these things are normal viruses getting made, and they're going to go off and then infect new host cells. Very, very rarely, like 1 in a million, though, the phage proteins, instead of wrapping around a copy of the phage genome, the phage DNA, they make a mistake and they wrap around a copy of one of those digested bacterial DNA fragments. So remember, at one point you digest the host chromosome and the DNA is in fragments. Very, 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 very rarely, doesn't happen very often, but those phage proteins wrap around bacterial DNA instead of viral DNA. And this creates what is then called a transducing particle. And there could be a few genes to a few tens of genes on that one. But it could be any random digested fragment of that bacterial DNA chromosome, any general fragment. This creates what is called a non-lytic transducing particle. If this goes off, and if this then comes in contact with a new different cell, so this is going to be my new cell, this is going to be a recipient cell. Again, what happened here is my donor cell, this was my donor cell in a transduction event. By the way, what happens to my donor cell? It's dead, it dies, but some of its DNA made its way into that transducing particle. If that transducing particle then comes in contact with some new recipient cell, what it injects, it doesn't have any viral DNA, it injects the bacterial DNA instead. So it transfers bacterial DNA, not the phage viral DNA. This then can get incorporated into the bacterial chromosome. By the way, since there is no viral sequences, this is not going to cause disease in the recipient cell. It's just going to inject the DNA, and that's all that's going to happen. There's no virus in chat here. It's a transducing particle only. This will then transfer that DNA, and that DNA can sometimes get incorporated into the bacterial chromosome. Those genes just got transferred in the recipient cell. The recipient cell might have picked up those new genes. This is referred to as generalized transduction because theoretically any random piece of chopped up bacterial DNA chromosome could get transduced. And it's kind of at random and in general how that takes place. Again, this is a mistake. It's a 1 in a million, but viruses are constantly infecting host cells, constantly making copies of themselves. And there might be trillions and trillions and trillions of viruses. There is more viruses. I think I said this at the beginning of the class. There's more viruses in a liter of seawater than there are humans on the planet. In 1 liter of coastal seawater, there's more viruses in that than there are humans on the planet. So there's viruses everywhere all the time. Most of them are harmless to us, like bacteriophages. But they can also do this because there's so many of them. Even a 1 in a million event takes place somewhat regularly because there's just so many viruses around. Does that make sense, everybody? Questions about that? All right, some viruses infect host cells. Oops. So there are some viruses that infect host cells and then immediately start to replicate inside the host cells, make copies of themselves, and kill the host cells. These are called lytic viruses. So some types of viruses immediately start reproducing, immediately almost kill those host cells while they're making copies of themselves. And these are referred to as lytic viruses. Lytic viruses can cause generalized transduction. And this is not something they're trying to cause. This is a mistake that happens sometimes. There is another type of viral reproductive strategy, though. Some viruses do what is called lysogeny, or the lysogenic cycle is another name for that. And this will come back up again when we talk about viruses and virology at the end of the semester here. Some phages infect bacteria, but they do not immediately lyse the host cell, and they do not immediately start to make copies of themselves. So what they do, my virus docks with my host cell. It injects its genetic material into the host cell. So again, this is the kind of injection step. Okay, but then lysogeny is when the phage DNA stably integrates and becomes part of the host cell chromosome. It can stay there dormant and hidden as something called a prophage. So again, here is a prophage. The bacterial DNA is in blue, just like it was previously, but my prophage, my viral DNA, has inserted and become part of the bacterial chromosome. And it can hide there and remain dormant for long periods of time. And there's a lot of different viruses that can do this. So that phage DNA will always integrate at a very specific DNA sequence called an ATT, or attachment site. So it always looks for this very specific DNA sequence and integrates at that same spot every single time when a virus infects that particular kind of host cell. But that integrated DNA is called a prophage. Okay, and you guys should know what a prophage is. Prophages are going to come up again. Sometimes they're also referred to as proviruses then as well. This is an alternative name for the same thing. What ends up happening, though, is that phage DNA hides inside of the bacterial chromosome. So it's there. But if the bacteria goes and replicates its chromosome, goes through cell division, now you go from one infected host cell to two infected host cells. If then these make copies of their DNA and go through cell division, now you've got 4 infected host cells. And in essence, the viral genome is hiding in the form of that prophage inside of there the entire time. And the cells replicate, and all of them have these hidden viral sequences in them. They can stay dormant for long periods of time, but eventually they can also wake up. And so what ends up happening is stress can cause them to transition from the lysogenic cycle to then the lytic cycle, where they then start to actively make viruses. Stress will then activate the phage lytic cycle. Why would stress activate the virus to, in essence, wake up and start making copies of itself? If the bacterial cell is under stress, stress would be things like high temp, low nutrients, other stuff. If this bacterial cell is under stress, what might happen to the bacterial cell? It might die. What happens to the virus if the bacterial host cell dies before it makes copies of itself? It also dies. So if you have a bacterial cell under stress, what ends up going to happening is that bacterial cell is in danger of dying. If that bacterial cell dies before the virus makes copies of itself, the virus is also dead. So under stress conditions, the phage wakes up, the prophage cuts out of the bacterial chromosome. It then actually starts making copies of the viral genome, copies of the viral protein, and eventually then it's going to burst. It's going to make copies of the viruses that then burst out and kill the host cell. So it'll transition from the lysogenic cycle, where it's hiding in the bacterial DNA, to the lytic cycle, where it's actively making copies of itself. Questions about that? By the way, there's several human viruses that also do this. Anybody think of a viral infection that can be dormant for long periods of time? Herpes. And herpes can do the lysogenic cycle. Herpes, the viral DNA from herpes will insert into your DNA sequences and hide there and can be dormant for long periods of time. Under stress conditions, could be lack of sleep, not eating well, could be you got an exam coming up. Then sometimes those herpes viruses wake up and then you get things like, for instance, cold sores or other stuff like that. And shingles is another example. If you've had chickenpox virus early in life, you can be vulnerable to shingles later in life. And what ends up happening is if you've had chickenpox, you actually still have chickenpox, technically speaking, but it's hiding as proviruses inside of your DNA. Specifically, it hides inside the DNA of your epithelial cells and nerve cells, for instance. Another example would be HIV. HIV, you guys probably heard that people can have no symptoms for long periods of time. That's because HIV will insert a copy of its chromosome, the viral DNA, into our chromosomes. And it can hide there and be dormant. When people have active symptoms, it's why the HIV is actively reproducing. Does that make sense? That's when it causes symptoms. All right, questions about lysogenic versus, say, lytic then. These are going to come back up when we talk about viruses. I'm going to go through them in a bit more detail then. How does that relate to then specialized transductions? Sometimes phages. Remember, what ends up happening is normally you have, for instance, a bacterial cell. What happens normally in that bacterial cell, again, you have then the bacterial chromosome. You've got that prophage that is inserted into it. Typically, when it transitions to the lytic cycle, it's going to cut here and here. So typically during the transition, sometimes the phage cuts out of the chromosome and then starts making copies of itself. Usually it cuts on one side of the prophage and then the other side of prophage, and that almost always happens right. Almost always happens correctly. But sometimes the phage messes up and say instead it cuts here and here, for instance. Sometimes it messes up the cut sites. Maybe it cuts in the middle of the prophage, but it also cuts and gets some of the bacterial DNA sequences. So again, if you had that bad cut over here, this is again my purple cuts, it leads to this type of situation. When the prophage excises out, it gets some of the viral sequences, but not all of them, but it also gets some of the bacterial sequences that are adjacent or next to the prophage. Could be to the left, it could be to the right, but it's always going to be next to the prophage integration site. This then will get packaged up into viruses and it creates transducing particles. These viruses will be these transducing particles that are carrying a mixture of both viral DNA and also bacterial DNA. But they missed some viral sequences, those got left behind. And because of that, those are not going to be able to make viruses in the future. This is bad for the virus if this happens. It's good for anything that might get infected by it, though. And so these are not going to be infectious particles. Again, these are going to be what are called transducing particles. In this particular case, what's going to end up happening is these will transfer that mix of viral and bacterial DNA sequences, but usually the viral sequences, there's not enough to make new viruses. Okay. And what is going to end up happening is it transfers those adjacent bacterial DNA sequences, and this is called specialized transduction. It's called specialized transduction because only genes near the ATT attachment site, near the prophage integration site, can be transduced. It has to be on one side of the prophage, but it has to be that specific part of the bacterial chromosome getting transferred, not any general fragment then, like in generalized transduction. Questions about that? Questions then about the different types of horizontal gene transfer, transformation, conjugation, transduction. All right. Then I have a pathogen of interest for you, which is going to be influenza A, and we're just coming out of flu season. So again, viruses are obligate parasites that must infect host cells in order to reproduce. There are a lot of different kinds of viruses, though. Your pathogen of interest that I'm going to go through is a type of virus called influenza A. This belongs to the Orthomyxoviridia family of viruses. Orthomyxoviridia family includes influenza A, influenza B, and influenza C. We're not going to worry much about C. Typically, flu season is caused by some type of usually influenza A or also influenza B virus. So what we call flu is either influenza A, and there's a lot of different kinds of influenza A, or a type of influenza B. Influenza A is then the most common cause of flu. Then. Especially early in flu season, it's almost always influenza A. Influenza B tends to hit at the end of flu season. Flus, there's actually many different strains, substrains of influenza A, and they're named after the types of hemagglutinin and neuraminidase proteins on their surface. And I'm going to talk about both hemagglutinin and neuraminidase here in a second. Symptoms from flu include things like fever, cough, headache, muscle aches, fatigue, chills, and sweating. Those are going to be typically the most common symptoms from this. Sometimes you also then get some upper respiratory symptoms. Okay. Prevalence for this one. It kind of depends on the flu season. It does vary a little bit. It's going to work. So this is basically just going to be kind of some statistics on different than flu seasons. This is cases of flu in the millions in the United States over the last, say, 13, 14 years. So sometimes we can have, for instance, flu seasons where we have about 21 million cases, 9.3, but it usually ranges anywhere from about 10 million cases of flu in the US per year to up to 40 million cases of flu per year in the US is kind of what's being shown. By the way, anybody happen to know why there were so few flu cases in 21, 22? Huh? It was COVID-19 was in the middle of the worst part of it. A lot of people wearing masks. Masks didn't work the best for preventing COVID-19 spread. COVID-19 spreads way better than flu. But it turns out masks work great for preventing the spread of flu, especially if you're wearing N95 masks or better, for instance. So a lot of people were masking. A lot of people were doing things like social distancing. It actually turned out that worked great for preventing the spread of flu. Unfortunately, COVID-19 is just way more contagious than flu, so it didn't work as well against COVID-19, unfortunately. We're going to talk about this later. Flu has an R0 value. R0 is a measure of infectivity. It has an R0 value of 1.5. That means it's for every person that gets it, they give it to 1.5 more people. COVID has a R0 value of 10 to 12. That means for every person that gets COVID-19, they give it to 10 to 12 more. Oh, yeah, nobody's quite sure about that one, actually. That was a weird one, and they're still trying to figure that out. Basically, the one thing that happened here, the flu vaccines that year just nailed it. The flu vaccines that year, they nailed what type of flu circulated around, and they were very, very protective against flu, much more so than normal. But it turns out the flu viral strains that were circulating this year were very, very, very similar to the ones from the year before. So people had a lot of herd immunity and immunity against it already. So it was 2 years in a row with very similar flu circulating. So a lot of people were already protected because they got exposed to it the previous year. So that's the thought process on that. Good question. Well, was there another question back there then? Okay, yeah, it was the variety of flu that circulated this year was also the variety that circulated this year. Usually they mutate and change much more. They didn't do that for some reason this year. Yeah, that's a good question. And I was actually I had to look that up myself when I saw the graph. Okay. So the CDC also keeps track of flu per year. So for instance, this is flu season. Flu season is going to be basically starting in around, what week are we here? Week 46. So this is going to be towards the end of the year. Week 52 is like the end of December. Week 1 is going to be beginning of January. But if we look at flu, we can see that the flu season, typically flu's pretty light as we go from summer to fall. As we then start to get into November, December, then we're starting to get typically flu seasons go. This is flu season from this past year, for instance. It was a very bad flu season, by the way. And so then by week 10 here, we're starting to get into March and the flu levels are now going down, for instance. Okay, but this is a keep track of the flu levels and everything like that. It was a bad flu year, which is why the map kind of looks so bad here in the middle. Okay. There is annual flu vaccines to prevent the flu, and those often work very well. They don't always prevent the flu, but they often make it so that if you do get vaccinated and you do get the flu, it often makes it so the flu is less severe and the symptoms aren't as bad then as well. So the reason to get the flu vaccine is to actually make for less severe flu cases or to prevent the flu entirely. The more people that get the flu vaccine, also the harder it is for flu to spread to the population. So with more people vaccinated, the flu doesn't spread as readily. Unfortunately, in my experience, college students don't often get the flu vaccine, and then the campus just gets demolished every year with flu, and it spreads to the dorms like a wildfire. But that's a whole separate story. The other thing is flu is a type of then RNA virus. RNA is less stable than DNA. By the way, some viruses are DNA viruses, like chickenpox or herpes or hepatitis. Those are all DNA viruses. Some viruses are RNA viruses. And this would be, for instance, things like flu, COVID-19, HIV are all RNA viruses. DNA viruses have a DNA genome. RNA viruses have an RNA genome. RNA is less stable than DNA and has higher mutation rates. That means these viruses change faster. Because the viruses change faster, it's harder to develop a vaccine against them. So the reason you have to get an annual flu vaccine is because the variety of flu that's circulating this year, that flu will change, mutate. And the variety of flu that's going to be circulating next year is not going to be the same. And our immune system's not going to recognize it anymore. So you guys probably heard of COVID-19 variants. COVID-19 or the Delta variant or the Omicron variants and all that same thing. It's a mutation causing changes over time. It's evolution. And so what ends up happening here is then the RNA, it changes over time, the virus changes over time. And so last year's flu vaccine is good against last year's flu, but not against this year's flu because it's changed. And so you do have to get different flu shots every year to keep up with the changing viruses. Questions about that? Sometimes they do a good job of making the flu vaccine, but they have to predict 6 months prior to flu season what the predominant flu strains are going to be, and then make those vaccines 6 months beforehand. Sometimes they do a good job predicting it, and they have good flu vaccines for that season. Sometimes they do a bad job predicting it, and the flu vaccines don't work as well. Does that make sense? They go constantly, the CDC, other governmental institutions, also just some healthcare environments, they're constantly monitoring what kinds of flu are circulating around, using that to predict what is going to be the predominant strains in 6 months or so. Sometimes they do a good job, and like the vaccines are like 60% effective. Sometimes they don't do such a good job, and they're maybe only 40% effective. Influenza A, there are different kinds of influenza A. Hemagglutinin. These are viral proteins that allow for the virus to attach to host cells and then enter into the host cell. They recognize the host cell and allow the virus to then enter the host cell. Neuroanamidase. This is a viral enzyme that's necessary for the release of replicated viruses from the host cell. So the hemagglutinin helps viruses get in the host cell. The neuraminidase helps the virus get out of the host cell. Both of these are necessary for viruses to get big. And because these two proteins are very, very important for viruses, it actually turns out also different types of influenza can have different versions of either hemagglutinin or neuraminidase. There are 16 different types of hemagglutinin and 9 different types of neuraminidase in flu A. And these are recognized in the immune system. They're central to viral reproduction. And so a lot of the flu strains are named after them. So there's H1 through H16 and N1 through N9. And you might have heard of different kinds of flu strains like H1N1 flu, H5N1 flu, or anything like that. But you have different types of flu will do a different hemagglutinin molecule or a neuraminidase molecule, and sometimes the flus mix and match these. All influenza A is capable of infecting bird populations. Actually, flu comes from birds originally. All flu circulates around in bird populations. Some affect pigs and humans and other mammals. I kind of have it mapped out here. But ultimately, aquatic birds, especially waterfowl, are going to be one of the more common reservoirs for influenza. There are different kinds of influenza A. For instance, H1N1. This is caused a pandemic in 2009 and 2010. This was a version of flu that had H1 type and N1 type for its hemagglutinin and neuraminidase. This was estimated to cause somewhere between 43 to 89 million cases worldwide. It did circulate very readily through human populations. It was estimated to cause only about 284,000 deaths worldwide. I say only because that's a lot of infections and not very many deaths. Turns out H1N1 probably started off in pigs in humans, in Mexico, then jumped into humans and then started circulating around through human populations. This was a novel strain of flu, a new strain of the flu at the time, so no one had any immunity or herd immunity to it. We're going to talk about herd immunity later in the class. But this means it spread through this. H1N1, though, ended up having very high transmission rates in humans, but very low virulence. What that means is it spread real easy, but it had very, very mild symptoms. So they were very concerned about it at the time, but it ended up having such mild symptoms, they didn't do much about it. Does that make sense? So got a little lucky there. The reason I say we got a little lucky is because they were very concerned about it because the Spanish flu, this was actually, you might have heard of the Spanish flu. This caused a pandemic in 1918. This was also caused by an H1N1 variant of the flu. But this one infected 500 million people across the world in 1918, and it killed 50 to 100 million people by estimates then. By the way, that is a mortality rate of anywhere from 10 to 20%. So this one was very transmissible and also had high virulence, so was very deadly then as well. So they were really concerned when H1N1 came out because it looked similar to Spanish flu. We got lucky it wasn't, in essence. You might have heard of bird flu in the news quite a bit. Bird flu is a type of influenza A that is H5N1. So bird flu, when you see that in the news, and there's a lot of that in the news over the last couple months, this is going to almost always be in birds, although it seems like it's hopped into cows lately on farms then as well, which is a new development. H5N1 there is been 70 cases of H5N1 bird flu in humans this past year. However, almost all of those were from humans being exposed to either infected dairy cows or infected poultry, mostly chickens. There is no transmission between one human to another human for bird flu currently. So it just does not transmit from human to human. You almost always get it from contact with an infected animal. If they find it in cow herds, or they find it, well, maybe cow herds, but if they find it in like chicken farms, they kill off all the chickens on the farm. And if you guys are wondering why eggs are so expensive, this is why, actually. There's been several outbreaks of bird flu in chicken farms this year. And when that happens, they kill off the entire flock because it's too hard to test all of them. And they do not want the workers or they coming in contact and catching bird flu. They also don't want bird flu to actually learn or evolve so that it starts hopping into humans and spreading. The reason they don't want it spreading in humans is because bird flu in humans has a mortality rate of 40 to 50%. So if bird flu was to hop into humans, nearly half of all humans that catch bird flu from an animal end up dying from it, even with medical treatment. It has a very high virulence, and it has a very high mortality. So they're killing off anytime they detect it, they're killing off all the animals that might have been exposed to limit exposure as much as possible and limit it kind of transferring into human populations as much as possible. Questions about that? Unfortunately, there's been a lot of outbreaks of bird flu in chickens, which is why also they have to kill off all the egg-producing chickens. That means less eggs, and that means eggs is more expensive if you're shopping in the grocery store. It's been a particularly bad year for that. All right, questions about that or flu? All right, biofilms is the next thing. Biofilms is also going to be our transition to most of the rest of the class is going to be on disease. Bacteria, and probably the way you were taught about bacteria in high school is that they are unicellular. They aren't. That they are often planktonic or free-floating, that they're very independent, very individualistic. And this is often the way it's still taught, and it's often incorrect as well. The more modern view of bacteria is that bacteria often grow in loose multicellular arrangements. It's not a true multicellular organism, but they like to grow in groups. They like to be next to their neighbors. They like to be cooperating with one another. That they don't like to float in liquid. They actually like to be surface associated and anchored onto some type of surface. They don't like to be free-floating cells. They like to be attached to something, a substrate or a surface. That usually they're cooperative. They talk with one another, communicate with one another, cooperate with one another, and they prefer growing in these group formats. And there's a lot of advantages for them growing in a group. So for instance, they can do things like social motilities. For instance, we talked about swarming, for instance, as a type of group or social behavior. But we're also going to talk about social behaviors called biofilms here today, just a little bit to introduce that topic. But then also we're going to hit this in more detail in Friday's lecture. Then, biofilms is what we're going to talk about. Definition, my definition of biofilms. You will find some alternative ones online if you do a Google search, but the definition of biofilm is a multicellular aggregate of bacteria held together by extracellular polysaccharide layers, EPS layers. There's kind of two parts to that definition. There's the multicellular grouping and the EPS part. And I'm usually looking for both of those parts in any kind of definition. Technically, a lot of times colonies on a plate are kind of a version of a biofilm. We can have pellicles growing at the top as a type of biofilm. A lot of times with biofilms, though, we're talking about bacteria on a surface-water interface, a surface-liquid interface then as well. Okay, and this is just an example then of a biofilm. Each individual thing is a cell. So for instance, this is one cell, but a biofilm is going to be a big grouping of cells. This is my surface right here, and we're kind of looking at a cross section down through, but this is in essence a tower or group of cells that's anchored onto that surface, and all those cells are anchored and attached onto one another into this loose multicellular arrangement. And that's what we're typically talking about with biofilms. Questions about that? Okay, next lecture, I know I'm out of time. So next lecture, we're going to talk about biofilms. We're going to talk about how biofilms develop and form, some of the things that control it, but we're going to talk about why they grow as biofilms, because there's a lot of advantages to growing in a biofilm structure. Don't forget then, there is a quiz to take today. It's not going to be open up for about 10 or 15 minutes. I got to modify it just a little bit, but it'll be open until 5 p.m. tomorrow.